Hi, it's my pleasure to introduce Adam Guss as our speaker today. He's a genetic and metabolic engineer at Oak Ridge National Laboratory in the Biosciences organization. He started out life in Missouri, went as an undergraduate to the State University at Rolla, got his PhD from the University of Illinois at Urbana-Champaign, where he worked in microbiology, studying electron transport pathways used by members of the archaea to produce methane. Archaea, primitive organisms. I saw recently that BP purchased a company called Archaea, which is supposed to be a big property in the synthetic biology arena. He subsequently went to postdoc at Harvard, where he studied phylogenetic and metabolic diversity of rarely cultured and non-cultured bacteria that were present in the lungs of cystic fibrosis patients. He then went on to work at Dartmouth, doing postdoctoral research on genetic engineering, metabolic engineering of microorganisms for producing biofuels from cellulosic biomass. Continues in similar areas here at Oak Ridge National Lab. I don't remember how long we've been there, but it's
12 years or so.
Okay. We were neighbors in building 1505 for a number of years. He's been working on using genetics and synthetic biology to engineer microbes to convert plant biomass and plastic waste and other materials into liquid fuels and other value-added products. Important stuff for our future. Looking forward to hearing what you have to say.
Great, thank you very much, Ellen. Perfect. Okay, so first, thank you very much for the invitation. It's a real privilege to be able to tell you all about the research that my group does. I know you said no questions until the end. If that's a hard policy, that's fine. But
no, that's okay.
Okay, so I'm going to try to keep it at a level that that physicists and other people can understand. But if you want more information or need clarification, by all means, I'm happy to slow down and clarify things. We all know that climate change is a major issue facing humanity right now. And if we don't stop our addiction to petroleum, then it's going to be bad. And so we really need sustainable fuels and chemicals to displace the petroleum that we use today. And it's going to require an all of the above kind of strategy in order to make this happen. So we need wind and solar, obviously, and electrification of personal vehicles and other things like that. But when we start looking to the future, and there are certain classes that we're not going to be able to electrify. So we're going to need liquid fuels for aviation fuel. We're not going to have battery powered jetliners or military jets. And the same is true for marine transport. So we're going to need liquid fuels for the foreseeable future. Also, pretty much all chemicals that we use are derived from petroleum. All of the polymers that you see around us, they're essentially all of it is derived from petroleum. And so if we want to displace petroleum and fossil sources of carbon, we need to replace those chemicals too. And so things like lignocellulosic biomass and other renewable feedstocks and waste resources are likely to have an essential role to play in humanity's future, even with things like electrification. And just to give people kind of a feel for the scope of the problem, in 2019, in the US, we consumed about 20 million barrels of petroleum per day. And just under half of that is used for gasoline. And about 2/3 of it overall goes to the transportation sector, so including things like air travel and marine. And so, when you add that all up, that's also almost 150 billion gallons of gasoline per year. And then all of the associated, amounts of jet fuel and so forth. Plastics are also a major issue. I don't see too many water bottles around the room, but we use plastics everywhere. And this is just kind of a summary figure of the total amount of plastic that humanity has produced since about the 50s or 60s. 8,300 million metric tons of plastics have been produced. A good chunk of that is still in use, but the majority of it has been discarded, and only a tiny fraction of it has been recycled. And so ultimately, all of that carbon ends up in the atmosphere, or polluting our waterways and so forth. And so we really need ways of taking current plastics and not just throwing them out into the environment. And then we also need to envision next generation plastics that are infinitely recyclable, recyclable by design, but also biodegradable for those times when they do make it out into the environment. And there are a lot of different political opinions out there on how important this kind of thing is, but regardless of where you stand politically, the need for renewable fuels and chemicals is really important. So if you care about mitigating climate change, switching to things like cellulosic biomass as a feedstock is really critical for displacing petroleum. But if you care about rural development, rural economies are going to grow as we switch to more and more renewable feedstocks. And then for people who care about national security, we import a lot of oil from foreign countries, and a lot of those countries don't really like us very much. And so separating ourselves from that dependence on foreign countries, being able to grow our own energy is going to be really critical for increasing national security for the future. So I've been at Oak Ridge for about 12 years. I really love it here. And one of the reasons is that we get to do big science. We get to try to solve the big problems that a single academic lab, even at Harvard or Berkeley, is not going to solve on their own. Things like, how do we make a renewable bioeconomy? These are not going to be solved by individual people. And so the Department of Energy is really good at bringing together diverse groups of researchers to solve these big problems. And so my group gets a lot of funding both from the Office of Science and the Bioenergy Technologies Office, which is from the Energy Efficiency and Renewable Energy side of DOE, which is the more applied side of DOE. And so we have projects like the Center for Bioenergy Innovation, where we're trying to develop organisms that can eat lignocellulosic biomass, so the non-food part of plant material, and make fuels out of it. And then we're also in a project led by Pacific Northwest National Lab. We're trying to understand how can we engineer microbial communities to help support plant growth and make our agriculture more secure from various potential attacks. And then on the more applied side, we have the Agile Biofoundry, which is a consortium of national labs working to make the process of bioengineering faster, higher throughput, more automated, so that we can accelerate the engineering of microbes to help enable a bioeconomy. We also have the Bottle Consortium, which is an acronym for a big long title. And there we're trying to both break down modern plastics, and then what my group does is use microbes to upgrade the breakdown products to make next generation plastic building blocks. And then we also have a new consortium focused on utilizing CO2. So a lot of times the CO2 would come from bioethanol plants. So when yeast or other organisms ferment sugars into bioethanol, there's a lot of CO2 that's released. So companies can capture that CO2, and then we're developing the technologies to use microbes to reduce that CO2 into renewable chemicals. And then within the EERE space, We also have a lot of opportunities to team up with different industrial partners to help them solve their big problems in biotechnology. And so kind of for all of these reasons, I'm really excited to be here at Oak Ridge and doing this kind of big science research. So just to kind of bring us all onto the same page in terms of biology, my work really focuses on genetic engineering. And so there's what's called the central dogma of biology, which is basically at a high level how information transfers within biology. So you probably all know that our genetic material is made up of DNA, and so there's a process called replication where cells just make more copies of that DNA so that they can divide, and so that each cell gets a copy of the DNA. And so we do a lot of work with DNA, either taking DNA out, putting DNA in, and making additional copies of DNA to manipulate how the cells do things. But the DNA itself isn't active. It's the kind of code that tells the cell what to do. And so the DNA gets converted into something called RNA. It's a process called transcription. And then that RNA gets decoded into proteins. And proteins do the vast majority of the work within your cells. And so most of my work focuses on manipulating the DNA, but with the purpose of changing the proteins that are there. Okay, and so as we engineer these organisms, one of the first things we need to do is choose what organism do we work with. And the vast majority of metabolic engineering research that's been done historically and even today is both in academia and in industry is performed in what are called model organisms like E. coli or yeast, Saccharomyces cerevisiae. And the reason is because they're really, really easy to work with. It's really easy to take DNA out and put DNA in, and we know so much about their metabolism, how they regulate genes, how the different genes control what metabolic pathways are used, and therefore they control flux through carbon metabolism. And we just understand it so much better, especially in E. coli, but also in Saccharomyces, so much better than we do in any other organism. But the phylogenetic and the metabolic diversity of microbes is truly vast. So on the left side here, and you're not really meant to be able to read any of this, but the left side is a phylogenetic tree. So it's essentially a family tree of a fraction of the bacteria that are out there. And so like any of these different colored groups, they're probably more different from each other than we are from plants or fungi or amoebas and whatnot. So there's a lot of diversity out there. And then the right side is kind of a representation of carbon metabolism. So as sugars and fats and things get converted into other molecules within inside a cell, each of those nodes is a unique molecule, and each line is an enzyme that does interconversions. And even that is just a small fraction of what microbes can actually do in terms of converting one chemical into another. And E. coli and Saccharomyces yeast represent just a tiny fraction of that diversity. And there are a lot of organisms out there that do really cool things that are really challenging biochemically to do. So things like the extreme pH tolerance, so growing at pH 10 or 11, growing at pH 1. There are organisms that love to grow at pH 1 and 60 or 70 degrees Celsius. And that's where they grow. And so if you want to have a process that you've engineered, if it makes sense to have that process happen at pH 1, you're never going to get E. coli to be able to do that. You really need to be able to work in some of these more non-traditional organisms that do that natively. Because if you were to engineer E. coli to grow at pH 1, it would just be so fundamentally different. You would need to change pretty much everything inside the cell, and so it wouldn't even be E. coli anymore. And so the question is, why don't people, I guess other things that are important, things like cellulose utilization. So growing on plant biomass, plants have evolved to be really hard to break down, but there are certain microbes that are pretty good at it, but it's not E. coli and it's not yeast. And so if we want an organism that's really good at breaking down plant biomass, we need to go find an organism that already does it well and then try to engineer it. So then the question is, why don't people just do this all the time? Why do people even bother with E. coli and yeast? And the answer is that in most cases, we don't have the tools necessary to do genetic engineering in those organisms, and we don't know much about their biochemistry. And so even when we can manipulate them genetically, we often don't know what to do. And so the genetic tool side of things is one of the major barriers to being able to manipulate these organisms, to engineer them to do things for biotechnology. And the first step is just getting DNA inside the cell. So that's one of our major challenges, and something that my group works a lot on. And so kind of thinking about the ability to do genetic modifications in organisms kind of more broadly, you can also think of genetics as a type of logic. So if you delete a gene, then you can ask what happens to the cell when that gene is no longer there. And so based on that difference between what the original cell is able to do and what your mutant cell is able to do, you can infer the function of that gene. You can deduce it. And the same thing with adding in a foreign gene. You add a new DNA in, and then you ask, what is the cell capable of now that it wasn't capable of doing before? And so genetics is a way of helping us really understand biology much, much better. And so if we could just broadly start genetically modifying lots and lots of organisms, it would really open up lots of new fields of research, not only in the bioengineering type of things that I do, but also in areas like medical microbiology, understanding how pathogens work, discovering biosynthetic pathways for antibiotics. And then, things like looking to the future, engineering whole microbiomes, so whole communities of microbes, maybe engineering the microbes that are in your gut to do certain things as potential applications in the future. And so I'll talk about the work that we do in a couple different areas. So first is just developing technologies for doing genetic modification in different organisms. And then one specific tool that we're helping to pioneer that makes it really easy to insert foreign DNA into an organism's chromosome. And then I'll talk about a few vignettes of the research that we're doing with things like plastics, like growth on plastics, and growth on sugars, and conversion of lignin, which is a more challenging part of plant biomass to break down because it's polyaromatic. Okay, so developing genetics for non-model microbes. So when we want to do genetic engineering in an organism, there are a number of steps that need to happen. So first, well, you make your DNA that you want to use for the engineering, but then when it's time to put it into a non-model microbe, first thing you need to do is get the DNA physically inside the cell. And it turns out there are a lot of techniques for this, things like an electroporator, which is this device here, which basically forms an electric field that kind of punches holes in the membrane, and we don't really understand it beyond that. Maybe the DNA floats in, we don't know, but the DNA gets inside the cell, and so that's one of many ways of getting DNA inside cells. The next step, though, is that it needs to not get degraded. So microbes are under attack in the environment all the time from hostile DNAs. Things like viruses, but also other things called like conjugative transposons and other classes of hostile DNAs. And so they've evolved ways of degrading that DNA as it comes in. And so it also tends to degrade the DNA that we try to put in. And so we need to address these defense systems. And then after that, you need ways of maintaining the DNA inside the cell, and also finding the cells that actually have gotten the DNA, because it tends to be a low efficiency event. But one of the biggest barriers is that second step of avoiding the defense systems, and that's something that my group spends a lot of time working on. So surely you've all heard of CRISPR. CRISPR is like an adaptive immune system. So when a virus or something like that infects a cell, it has the machinery to basically copy a section of that virus, and if the cell happens to survive, then anytime a similar virus infects it, it's like, oh, I recognize that piece of DNA, I'm going to go cut it up. So it's not a general defense, it's a very specific defense. But the DNA that we're putting in, the cell has never seen before. So CRISPR systems are not actually a big deal for us to avoid as we're doing this engineering. The bigger issue is called restriction modification systems. So basically the cells have enzymes that chew up DNA that is foreign. And so you might wonder, well, how does it degrade, how does it recognize foreign DNA versus its own? And it does that by sticking a methyl group, usually, on its own DNA in very specific sequences. So there might be a 4 or a 6 base DNA sequence where the cell always sticks a methyl group on it. And so if that methyl group is there, the enzyme doesn't degrade the DNA. But there are lots and lots of different systems in all of the bacteria. And so a virus coming from some other cell probably does not have that specific sequence methylated, and so the enzyme chews it up. And so our DNA gets chewed up a lot by these types of enzymes. But I just told you that the cells methylate their own DNA to protect their own DNA. We can do what's called methylome analysis, which is basically looking at all of the DNA bases that are methylated within a cell. And then that basically tells us what sequences need to be protected to not get degraded by these enzymes. And so technologies have been developed over the last, maybe 5 or 10 years that allow us to very quickly and easily determine which bases are methylated. And so then we can take the enzymes that do that methylation, put them into E. coli, and then we've engineered E. coli to mimic the methylation of our target organism. And so this allows us to bypass the defense systems that our chosen organism has evolved. So then the other thing is we need lots of genetic parts in order to be able to do rational metabolic engineering. So I told you the DNA gets replicated, obviously, to make new copies of itself. So that first column is just a lot of different sequences that we have in-house that we can use that tell the DNA to be replicated in a certain class of organisms. I also told you we need to be able to isolate the organisms that have been engineered. And so for that, we mostly use what are called selectable markers, or they're also called antibiotic resistance genes usually. And so if that piece of DNA, if the antibiotic resistance gene is on our piece of DNA when we put it in, then if we add antibiotic, we kill all the cells that did not get our DNA. And so we have a collection of these resistance genes. And then things like promoters that tell different cells how much how much a gene should be expressed, so how much protein it should be made. Terminators that say to stop making RNA and so forth. So we have all of these genetic parts that we can deploy in lots of organisms. And we've done this in lots and lots of organisms, and I won't kind of belabor the point of what they are, but one thing that I think is really important for national labs is to help enable the science of others. So we definitely do our own science where we're trying to achieve different goals of bioengineering and so forth, but we're also looking to help other people do their science better. And so as we develop these tools, we look to partner with academics and industry to help them do their engineering better as well. So that's a lot of the fundamental tools that we use to develop the ability to do genetic engineering in an organism. But there's one tool in particular that we've really started to push in a lot of different organisms because it allows us to very efficiently insert our foreign DNA into the cell, into the host chromosome to allow very rapid engineering. I'll skip that. So the class of enzymes that we use are called large serine recombinases. That just kind of describes the active site amino acid and what it does functionally. And what it does is it's an enzyme that basically links two pieces of DNA together. But it's two very specific sequences. And so on the right, you can see that about 50 base pair sequence, starting with TGC, GGG, and then the one right next to it, when those two pieces of DNA are in the same cell, one particular recombinase is from a bacteriophage, a virus called Phi-C31. It'll basically just link those two DNAs together. And so it'll insert our DNA into that second site, which we had previously put on the chromosome. And it does this really efficiently. And these enzymes are really cool because they don't work just in bacteria or just in like a single species of bacteria, but it works in pretty much all organisms. And so people have started using these enzymes for engineering, even in like plant and animal cell lines. And so these are just really powerful little molecular machines that are really good at linking two pieces of DNA together. Okay, so everyone needs an acronym for the tools they develop. So we've called our SAGE for serine recombinase assisted genome engineering. It's consistent with a bunch of other AGE type acronyms for somewhat similar tools. And this is how it works. So I've shown you one recombinase and its DNA sequences, but there are lots of different recombinases that have their own unique sequences. And so we can just put kind of a landing pad in the chromosome of our target organism. And like one is specific for the phage, BXB1, and another one is for BT1. All of these different viruses. So we have a bunch of different landing pads, and we can insert DNA into each of those. And basically, this is how it works. We put in one piece of DNA that has the red recombinase gene or integrase over on the right side, and then the other plasmid has the whatever genetic cargo we're looking to put in, and then it just has this corresponding DNA sequence that's going to when that recombinase protein gets made, it just goes over and links those two pieces of DNA together. And it does this at very high efficiency, and we've done this in a lot of different organisms. And just to give you a feel for the efficiency, for instance, the BXB1 recombinase, it'll give us about 10 to the 7 insertion events per microgram of DNA. So we can very easily build a library that has like 10 to the 7 members. So we've just engineered 10 to the 7 different organisms with our recombination system. And this is in 3 different organisms. So using the exact same system so the first organism is really good at eating aromatic compounds, which is why we like it a lot. I'll talk about it a little bit later. The second organism is really good at eating CO2. And then this organism is an industrial workhorse that's good at eating aromatics and sugars and a variety of other things. And it's really robust against different toxins. And so basically we're deploying this tool in a variety of different organisms, and it allows us to do this engineering very efficiently. And so I talked about the genetic parts that we use in a lot of different organisms. So traditionally, when people are developing tools for an organism, they really focus on their one organism, and it can take years to develop the tools that they need. But because this particular system works in lots and lots of organisms, we can basically build these tools once and then very rapidly deploy them in lots of organisms. And so in this case, we're looking at what's called a promoter library. So basically, it's the genetic part that tells the cell how much protein to make. And so sometimes we want to make a lot of protein, sometimes we want to make a little bit, but we want to know how much we're able to make. And so we need to be able to characterize these promoters in the organism that we care about. And so because we can use this recombination tool, we can build a single library of promoters and then test it in all of the organisms that we care about. And so now we know we can control gene expression. So this uses a fluorescent protein as a proxy for gene expression. So the amount of fluorescence equals the amount of protein that's there. And you can see that there's like 3 orders of magnitude differences in gene expression from the different promoters. And so now we can really tune gene expression in all of these organisms using these promoters that we've made. And then because it's so high efficiency, one thing that I think is super cool is that we can start multiplexing this. So we can insert not just one DNA, but multiple DNAs at the same time. And I'll tell you how we're using this tool in just a little bit. But basically, we can very efficiently insert up to 3 DNAs into the chromosome all at the same time. And so if you think about a metabolic pathway that has 3 different genes, you can make a library of gene number 1, a library of gene number 2, a library of gene number 3, where they're expressed at different levels. Maybe you grab what are called orthologs, so like essentially the same gene, but from different organisms that might have different kinetic properties. And you can make a library and just pop them all in at the same time. And if you have a way to identify the best one, you can very quickly, like for instance, the ability to grow on a certain compound, you can very quickly pull out the best pathway from all of these combinations. And just like with the other genetic tools, we do this in a lot of organisms. We like collaborating with people to help adapt it to their systems. And so this is just a really powerful tool for doing the types of bioengineering that we're interested in. Okay, so I have a little bit of time to kind of talk about some of our different metabolic engineering projects. So how we're using these different tools to try to make industrial chemicals and fuels. So the types of things that we do at Oak Ridge really need to focus on cheap feedstocks. So what I like to say is, like, if you're making the next greatest pharmaceutical, the cost of your feedstock doesn't really matter. You can use the most purified sugar because you're going to sell your final product for like $1 million a kilo. And so your starting substrate, like if it's $1 a kilo or $50 a kilo, no one cares. But if you're trying to make something like a fuel that you're ultimately just going to set on fire, your feedstock needs to be super cheap. And so just as a point of reference, the whole process of growing a plant feedstock, harvesting it, transporting it to a biorefinery, grinding it up, maybe doing some pre-treatment, like some chemical treatments to it, doing a bioconversion, doing separations to isolate your fuel molecule, and then transporting it to whatever distribution center. All of that needs to be done for about 25 cents a pound. So about $2, about $2 a gallon, might be our target. A gallon is about 8 pounds. So that puts you at about 25 cents a pound for that whole process. And so you can't use purified sugar. And so we're looking at things like residues, like agricultural residues, the challenging parts of plant biomass, like lignin, which I'd mentioned before is a polyaromatic, waste plastics, waste gas like CO2, CO2 plus electricity, for instance. We need really cheap feedstocks, and those cheap feedstocks are really hard for microbes to eat. Then we need to do all this metabolic engineering for all these metabolic pathways within our bacterium to get these cheap feedstocks converted into the types of chemicals we want. Whether it's solvents or polymer building blocks, fuels, and so forth. And so this is the big challenge that we're facing in industrial biotechnology. So one of the main organisms we work with a lot of organisms, probably 10 or so where we're doing like real metabolic engineering, and probably 20 or 30 where we're doing a little bit of genetic tool development for other people. But one of our favorites is called Pseudomonas putida. And we like it a lot because it's really good at eating aromatic compounds, and it's pretty tolerant of different toxic chemicals, things like aldehydes and solvents and so forth. And it's pretty straightforward to do genetic engineering in it. The genetic tools are pretty straightforward. So that's the main organism that we use, but we use others as well, and I'll touch on a few of them. So I'll start with a little vignette on plastics bioconversion. So one thing we've done is engineered Pseudomonas putida to help break down plastic water bottles. So PET, polyethylene terephthalate, which is recycling symbol number 1. So PET is it's a repeating polymer of terephthalate and ethylene glycol. And so if you hydrolyze it, you just get those molecules back out. And there's an enzyme that does that. It's called PET-ACE, for an enzyme that eats PET. And so what we want to do ultimately is engineer an organism to make this protein, PET-ACE, and then also eat the terephthalate and eat the ethylene glycol and make some new product. For instance, Itaconic Acid, which is a polymer, a potential building block for next generation polymers. So when we started this work, there was only two known metabolic pathways for eating terephthalate. Like in all the literature, only 2 organisms had ever been discovered that could eat it. And neither of them were any of the organisms that we work with. But we knew that Pseudomonas putida, one of our favorite organisms, has most of the metabolic pathways, but it's just missing those first couple steps in order to eat terephthalate. And so what we did is that we used our recombination system to build small libraries in all the combinations of these different genes. So we made a couple different levels of gene expression for pathway number 1. It actually has this particular organism, E6, has a second copy of these genes that's almost identical, but not quite. So we grabbed that one as well at different gene expression levels. And then a completely different organism called Rhodococcus jostii has a third pathway. And so we make all of those pathways, and then terephthalate also needs to get inside the cell. So we also grab the transporter genes that would enable terephthalate to get inside the cell so that the cell can eat it. And we make combinations of that as well. And we made all of basically the whole combinatorial library. And it turned out that only one combination worked. So the metabolic pathway from Comomonas, actually either version of it, they're functionally equivalent, it turns out, and the transporter from Rhodococcus. So no other combinations allowed for growth on terephthalate. And so using our combinatorial approach, we're able to very quickly find which pathway actually functioned in our organism of interest. It didn't work very well though. So we used a process called adaptive laboratory evolution. And basically we just evolved the cells to do it better. So you can imagine that if all of these cells, they're growing but not very fast, if one of them happens to get a mutation that makes it a lot better at eating terephthalate, it's going to very rapidly take over the population because it grows way faster than everything else. And so this is a fairly common approach to solving these types of problems. And so you can see at the beginning, these different strains grew pretty poorly. It took about almost 2 days for them to eat 10 millimolar terephthalate. But just transferring it once and allowing it to grow again, we selected for strains that were able to come up in about 30 hours. And then after about 7 or 8 transfers, which I think was maybe about 30 generations or so, we were able to select for mutants that were able to grow up in about 12 hours, which is about as fast as Pseudomonas grows on glucose. And so now we have a pretty robust pathway. And we sequenced the genome so that we could figure out what the mutations were, so that we understand why it's better. It turns out it was the transporter. We just needed higher gene expression for the transporter. But now we have a strain that grows really well on terephthalate. This project is a collaboration with researchers at NREL. And so they had engineered Pseudomonas to eat ethylene glycol, the other half of PET, and to secrete the enzyme PETase that does the breakdown of the plastic bottles. And so in this particular manuscript, we basically pulled all of those parts together, and in fermentation work that was done at NREL, we were able to show bioconversion of this breakdown product from PET, which is basically like a single unit of the polymer. So the terephthalate with ethylene glycols on each end. We're able to convert that into another like next generation polymer building block called beta-ketoadipate, which can be used in advanced nylons. And we're able to make over 15 grams per liter using these engineered strains coming from this bis-hydroxyethyl terephthalate. And then the collaborators at NREL also took an actual plastic bottle and used chemical catalysis to break it down into this BHET molecule. And we showed proof of principle that we can take actual plastic bottle that's been broken down and also convert it into this beta-ketoadipate. And then that beta-ketoadipate can then be separated and used for polymerization reactions to build actual polymers that are both biodegradable because it has this nice oxygen group here that makes it much more bioavailable for enzymatic breakdown. And so now we have this polymer that's biodegradable, and it came from waste plastic. Turns out we can also make it from like sugars and lignocellulose and so forth. You know, as we try to go from kind of model plastics that you might buy from Sigma or Goodfellow, when you get into real world plastics, PET is not just PET. It's not just terephthalate and ethylene glycol. It can have co-monomers in there, and they can be at relatively high abundance. So another thing we do is we go looking for microbes to discover new metabolic pathways that are not known in the literature today, so that we can break down these co-monomers as well. And so, like, maybe we just go to a compost pile or some dirt or sediment or something like that, and then just use that to put into a bottle that has the only carbon source being this molecule that we want to break down. And if there's any microbe in that sample that can actually break down the chemical, then it just grows up. And then we can identify that organism, sequence its genome, do some transcriptomics or proteomics to identify the genes that are involved, and then move those genes into our favorite Pseudomonas putida and engineer Pseudomonas to eat these molecules. So it's a way of so we're both doing like novel science, discovering new metabolic pathways, but we're targeting things that we care about for biotechnology as well. So we're able to do both fundamental and applied science at the same time. And so with one particular molecule, one of these co-monomers, we hypothesized that it was a 3-gene pathway based on related molecules that we knew about. And so we isolated a bunch of organisms that grow on this molecule, sequenced their genomes, identified potential genes that might be involved, and then did this exact kind of combinatorial pathway assembly in Pseudomonas putida to try to figure out which one might be best. We took this entire library. I think I don't remember exactly how many combinations there were. So I think it's 6 times 6 times 5. So a couple hundred combinations. We had 170x coverage of this library. So either all or essentially all combinations should be present within our mixture of different engineered strains. And then we just used a DNA-based sequencing assay to determine which genes were present in the population over time. And so the back row here, you can see the abundance of each gene. So of enzyme number 1, it's not exactly an even distribution, but all of them are represented. Enzyme 2 and the potential transporter, they're all there. When we grow it on this molecule a couple times, we very quickly come down to a single copy of enzyme number 1 being pretty much the only version that's there. So that is clearly the best enzyme of the ones that we discovered. And the same with the transporter, it came down to one that really was the best of all of them. And then for enzyme 2, it turned out that there were 2 that were roughly equivalent. So basically we ended up with 2 strains, each of which had, a single enzyme 1, a single transporter, and then had either version 1 or version 3 of enzyme number 2. And so now we have a strain that can eat this molecule. And this took about a week of after we had all of the plasmids built, all the DNAs built, it took about a week to figure out which pathway was best. And so that's the power of these tools that we're developing. I'll go quickly because I don't think I have a whole lot more time. These tools also allow us to very rapidly prototype pathways in new organisms as we look not only for the best pathway, but also for the best organism to host that pathway. So galactose is a sugar that's present in hemicellulose in a variety of different plant species. And Pseudomonas putida does not natively eat it. But we engineered Pseudomonas to express this pathway from E. coli, or mostly from E. coli, to be able to utilize galactose. So what we did is we used our recombination system. We took exactly that same DNA that we built for Pseudomonas, and we put it into Corynebacterium, which is another, was one of the organisms I mentioned earlier that's good at eating aromatic compounds, and Cupriavitis, which is the one's good at eating CO2. So none of them natively eat galactose, but we just dropped this pathway in, and now each of them are able to grow. And we can couple this with evolution to then ask, okay, maybe initially it's best in Pseudomonas, but if we evolve all three of them, then which one is best? And that's work that's ongoing. So we can also do it for pathway optimization in a single organism. So actually, if you go back here, so the Corynebacterium one, on glucose, on sugars that it natively eats, it'll grow up in 12 to 16 hours. And galactose is very similar to glucose structurally, and also biochemically for pathways. And so the question is, why didn't this work very well in Corynebacterium? Oops. We can use our recombination system to answer that question. So we took the pathway that I just shown you, and then we use our recombination system to drop in the second half of the path. The first half of the pathway, the ERA AB. The second half of the pathway, the DGO-CAD, so that's 3 genes, DGO-K, DGO-A, and DGO-D. Neither of those had much of an impact on growth rate, I guess I should say. But what we're measuring here is optical density, so basically how much light goes through the tube. And as the cells grow, they block more and more of the light. And so as more light is blocked, that's indicative of more cells being present. And so it's a proxy for cell growth. So putting in all of these metabolic genes didn't really do anything, but when we put in an extra copy of the transporter, now it grows really fast on galactose. And so we've discovered that we didn't have enough, just like the previous example, we didn't have enough expression of the transporter. And so now we have a robust strain of Corynebacterium that can also utilize galactose amongst the other things that it can eat. Okay, and so do I have enough time?
Yeah, sure.
Okay, so I think this is the last vignette that I put in here on lignin conversion. So lignin is an aromatic polymer that is randomly assembled using like radical chemistry. And so because it's randomly assembled, that makes it really hard for biology to break it down, because you don't have like a consistent single bond. Like cellulose, it's literally just essentially infinite polymer of glucose molecules that all have a beta-1,6 linkage. That's all cellulose is. And so if you have an enzyme that can attack that linkage, you're done. To be fair, it's a little more complicated than that, but essentially that's true. With lignin, there are tons of different chemical linkages that make it really hard to break down. But if we want to make cellulosic fuels a reality, we need to do something with this complex aromatic polymer. And so we're trying to engineer microbes to be able to deal with it. And we want to make a variety of molecules, just like everything else, we want to make a variety of molecules from this cheap feedstock for replacing petrochemicals. So the one I'm going to talk about now is called Itaconic Acid. My chemist friends tell me that Itaconic acid is really cool because it has 3 chemical handles on it. So it's a diacid, which means that it's good for polymerization reactions, but it also has an olefin group hanging off of it, which means that if you use it in polymers, you can also use it for cross-linking within the polymer. So then polymer chemists can do really cool things with a molecule like itaconic acid. And it's made today by a fungus from probably too expensive of a feedstock, but it has a variety of applications, I think largely as a cross-linking agent in different polymers for a variety of different applications, different plastics and paints and resins and so forth. So itaconic acid is kind of a good exemplar molecule to make from different feedstocks, because it's literally a single enzyme that you need to introduce in order to make this molecule. So an enzyme called CADA, which takes an intermediate in the TCA cycle, also called the Krebs cycle, if you remember your high school biology. And so it's literally a single enzyme, and And so we're hoping it would be fairly straightforward. And then the other thing is, as we're breaking down aromatic compounds using Pseudomonas, things like paracoumarate or benzoate, the metabolic pathways for those molecules all funnel directly into the TCA cycle. And so it's funneling all of this aromatic carbon really close to the molecule that we're trying to make. So that's why we're excited about making itaconic acid specifically from aromatics from lignin. We developed a biosensor that basically allows the cell to control when it does gene expression. I'm not going to belabor the point of the engineering, but basically we use nitrogen starvation. So we give the cells a limited amount of nitrogen. Nitrogen is obviously essential for growth, and so it'll grow. Pseudomonas will grow until it runs out of nitrogen. When it runs out of nitrogen, cell division stops, and it changes gene expression to start doing other things other than making new cells. And so we use that signal that the cell natively does to turn on gene expression of our one gene pathway, CADA. So when the cells run out of nitrogen, they turn on this production pathway and they start making itaconic acid. And they'll keep doing that as long as there's carbon around. So this is just how it works. I probably shouldn't spend much time on it, but basically it uses a native promoter within the cell to express a specific polymerase that does the gene expression, but it's regulated in such a way that it only happens when nitrogen is absent from the cells. And we did a bunch of other metabolic engineering that I won't really get into, but you can imagine this process. You have a growth phase, and then you have a production phase. So you can look at the yield for the overall process, which is basically the total amount of carbon you put in relative to the total amount of itaconic acid that you got out. So that's the yellowish orange bar here for each of these different engineered strains that we made. But then you could also imagine a future scenario where we've done further engineering and the cell can just keep doing the bioconversion indefinitely while it's in this production phase. And so we're interested in just how well does it perform after growth is done. And so that's the green bars. Using these different engineered strains, you can see that they got better over time with additional engineering. And our best strain, this last one, had an overall molar yield of almost 60%, which is really high for a biological process. But that takes into account the amount of carbon that went into making new cells. If you look just at the production phase, our molar yield was almost 120%, where the theoretical maximum is 133%. Because of the number of carbons and stuff. So we're getting really close to the theoretical maximum yield during this production phase. And so future work really needs to focus on having that production phase just keep going as long as we can to be able to make many grams per liter of itaconic acid. In conclusion, genetic tools are critical for understanding microbes and also for the manipulation of the microbes for different applications. The recombinases are really useful for the engineering that we do, and the tools that we built really put us in a position to team up with lots of different people around the world to help them do their science better, and also to enable the science that we're doing. And we're applying these tools, like I said, to things like upgrading of plastics, cellulose, lignin, and so forth to make renewable fuels and chemicals. And just thinking about how these can be applied in the future, we're trying to do work on engineering microbiomes for plant health and security, taking mixtures of plastic breakdown products, drug discovery, and so forth. There's so many things we can apply this to, and we're just starting to scratch the surface. And so with that, I'd like to especially thank DOE for funding both the Office of Science and EERE. Lots of people have been involved in this work. We have a lot of different collaborators, and I'm happy to take any questions.
Yeah, I have a real basic question about this manipulation to form these microbes. Is it sort of start as a one cell at a time thing where you go in there and then hope that that new cell reproduces very rapidly so you get a lot of this new material? Or do you have some sort of device that can do millions of these cells at one time?
Yeah, so it's a bit of both. We start with a population of cells, maybe 10 to the 9, 10 to the 10 cells, and then we do our best to introduce the DNA into all of them. But typically only a small fraction of them are actually going to get the DNA and replicate it and stuff. What we'll do is we'll take that mixture where some of the cells have gotten DNA and we'll spread them on Petri plates that have antibiotics on them, where the antibiotic kills any cell that didn't get our DNA. Because our DNA has a gene on it that makes it able to inactivate the antibiotic. And so if the cell took up DNA, it's able to inactivate the antibiotic, and so it survives. And so on your Petri plate, this population results in a single cell kind of sitting on the agar plate and then growing into a population or a colony where you can actually physically see all of the cells there. And within a single colony, all of those cells are genetically identical. And then we can, further work with it to validate that it is actually the mutation that we want and so forth. Did that answer your question?
Yeah, so the actual insertion, I picture, almost like going in like a surgeon into a single cell and doing something, but there's some sort of chemistry or something that allows you to do this multiple cells thing.
Yeah, so we can't really work with single cells very well. That's not literally true. There are devices where you can do like microfluidics and do the same kinds of things that we do, but literally on single cells. But that's not typically the way people do it. Typically, people will try to get as much DNA into as many cells as possible and then kill off all the ones that are wrong. I think you here,
Okay.
first.
First, I volunteer my compost pile and my recycle bin, anything you can use.
Perfect.
But seriously, have you and your industrial collaborators taken this really good basic set of knowledge into an industrial process at scale?
So not at scale. So one of our industrial collaborators is trying to scale up their process. We worked with a company to develop genetic engineering tools for their favorite organism, which had 0 tools when we started. And we developed the ability to take DNA out, put DNA in at high efficiency, and basically get rid of all the pathways they didn't want anymore. And then their whole process is making a biopolymer to replace polyacrylate in things like baby diapers. So to have a biodegradable, renewable, super absorbent polymer for various applications, but baby diapers is probably the flashiest one. So they're in the process of trying to scale that up and improve the process along the way. I don't remember exactly how large the runs were, thousands of liters, I'm pretty sure. But they actually purified, their polymer and made prototype diapers at a facility in France, I believe, somewhere in Europe. They're in the process of scaling it up. I actually have one of these diapers as kind of a souvenir because I think it's pretty cool. That's actually that's probably the company that I'm most excited about in terms of the ones we've worked with, just because it's such a great idea what they want to do. It's such a great technology, and they're making they're not like selling it on the open market yet or anything, but they're very much like pushing to scale things up and try to make a commercial process out of it.
Well, it's taking it a long way. That's great.
Yep. Some of the companies we work with, they are more technology companies, and they're looking to license out their technologies for other people to scale up. that's pretty common for early stage R&D startup companies in biotech. We're also talking with big players in the field to try to help them solve their problems as well. And so yeah, so we're doing our best to have an impact on things. And one of the things that the Agile Biofoundry really cares about, one of these large consortia that I'm a part of, is like understanding scale up. So like as you go to higher scales, things change. So like the amount of pressure in a 1,000,000 liter fermenter is very different than the amount of pressure in a 25 ml shake flask. And so that's just one thing, like the CO2 concentration, when you have all of that water, on top of, the pressure up top versus down below is different. There's just so much that can be different as you scale something up. And my group doesn't do a whole lot in that space, but for the project at large, and people at NREL and Lawrence Berkeley and other groups, we're really trying to understand like what's going on as you scale up to help companies be able to do it more rationally.
Adam, I'd like to switch to some of the questions that are in chat. The first one up there on the screen is there a role for increasing domestic natural gas and oil production, e.g. fracking, pipelines from Canada, et cetera, to alleviate energy security concerns?
So, I mean, I guess the short answer is yes, in the most literal sense. As someone who cares about climate change, my hope would be that we don't go down that road, or at least minimize the amount that we go down that road. But that's largely a political and economic question, not a technological one. So yeah, my hope would be that we don't do that any more than we absolutely have to. But yeah, it's certainly on the table, as are any other technologies.
A second question below that is 42% of TVA's generated electricity comes from just 3 nuclear power plants. Why is nuclear not mentioned as the best base load alternative?
Yeah.
So that Adam's work is on electricity.
Yeah, so I'm probably not the best person to answer that question. I do know that there are various perspectives on the importance of nuclear energy going forward. I don't feel that I'm really qualified to talk about it. From my perspective, it's a source of electrons that we could potentially, whether it's water splitting or whatever else, you make electricity, maybe you do water splitting, you get electrons. Biology can actually use those electrons. That's work that's, not my group, but there are certainly people working on bio-utilization of electrons from electricity, or things that have been produced from electrons from electricity. So like catalytic production of formate or other reduced CO2 compounds using an electrolyzer, for instance. So these are all things that are not really in my wheelhouse, but they're things that we try to interface with as we think about how can biotechnology potentially use some of these options as a feedstock for making next generation fuels and chemicals.
Well, the third question then on that list is, at the risk of sounding like a bad science fiction movie, are certain protections required for your lab to ensure that modified cells do not escape into the environment? Yeah,
it's a good question. So it's an active area of research for lots of groups around the world, including mine. So pretty much anything we work with, it's not able to eat like a hunk of plastic, for instance. We actually use chemical catalysis to break it down into soluble components that the microbes can eat. So they're not literally eating like a plastic bottle. And even if they did, there are natural organisms that have evolved to eat things like PET. That's actually how we found the enzyme PETase. Is that a group of researchers went to a plastic contaminated area and found some organisms that could eat polyethylene terephthalate, and they did some work and figured out the enzymes involved, and and so forth. And so, but these are things that take, years and years to break down in the environment. So in terms of actual technologies for prevent, well, I guess I should start. Everything we do, we follow like strict protocols for like biosafety and stuff. If there are any potential concerns about pathogenesis or plant pathogens or any concern where a reasonable person might say, well, is that okay to do? All that has to go through a biosafety review. Actually, everything we do has to go through biosafety review, but anything where there's any question goes through an enhanced review. Most of the stuff we work with, a lot of them are only able to work at higher temperatures. So we work with organisms that for like the ones that break down lignocellulose, they don't grow at Earth temperatures, at least current Earth temperatures. So like the lowest temperature that our organism can grow at is about 48 or so degrees Celsius. And it's really happy around like 60 degrees Celsius. And so it just cannot grow on an actual plant, for instance. And so we're also investigating genetic ways of minimizing or preventing spread in case of an accidental release. You can imagine a 1 million liter fermenter. If you have engineered organisms, even though we do our best to make sure that there's no realistic possibility of them being harmful if they were to escape, we still also try to build in mechanisms that make them die if they escape. So where they require some sort of chemical that only we add in the lab, such that it dies once it gets out into the environment, or maybe it's not able to make certain amino acids, or other things like that. And so like biosecurity and biocontrol are like major areas of research that the Department of Energy is very interested in, and pretty much any project that DOE funds that has bioengineering in it requires some sort of like biosecurity and biocontainment kinds of research associated with it.
The fourth question up there from Jim Rome is bio-based reactions are inherently slow. This problem has already been solved at an industrial scale by Proton Power in Knoxville. And you can visit the Wampler sausage factory in Lenore City to see it in action. They can use any waste without chlorine in it to produce fuel, carbon, 30% graphene, hydrogen and CO2, still net negative. So why spend money on bio approaches?
Yeah, so I don't have information on this particular company. But the fact that it's not a multi-billion-dollar worldwide company makes me presume that they're still trying to make the technology work economically. I hope that's not a bold assumption. And so basically none of these technologies work economically today. Bio doesn't, except for a few specific applications, like the Tate and Lyle plant that was in Lenore City, making propane diol for different polymers. That worked so well that it eliminated the equivalent petroleum-based process, just because it's so much better. But for most applications, there just is not a renewable or sustainable option right now, and so we need to fund all of them to try to find solutions that are actually going to work.
You really need to go. You need to go visit them in Lenore City, because they have $1 billion plant that's being built in Singapore. And they have it on a huge cattle farm in the West to process manure. And you really, I think they've solved the problem.
Okay, so I only heard part of that, but I think the person said that they're building plants in a variety of countries.
That's
and
$1 billion company, you said.
Yeah, yeah. So there have been quite a few catalytic based companies that have said the same thing and then crashed and burned. Same with biology, bio companies also. So I'm not trying to single out chemical companies. And so, you know, people have to start scaling before it's economical to learn how to get to large scale and to improve the process. You know, maybe they will actually take over the world and become the next, you know, $1 trillion company or whatever. If so, let's see. So this is fuel. So we'll still need chemicals. And so unless they're also making all of the chemicals that come from petroleum, then we'll still need bio for that. But until like Exxon and BP are out of business and displaced by this particular company, I think it's important to investigate as many different technologies as we can afford in order to try to make an impact on the future.
Some related questions that came from the feed, and I think that we all are having, is how far away are we from being on a path of actually using this technology to produce liquid fuels on a commercial basis?
Yep. So there are lots and lots of different specific technologies, and so any given one is at a different place. So if we're thinking fuels in particular, there are companies like a company called Lanzatech, which uses microbes to ferment carbon monoxide. So when that carbon monoxide, syngas is what it's often called. So when that syngas comes from steel mills, where it's essentially free, or even they get paid to take it, they're making bioethanol that they're selling on the market today. So they're making a fuel, and then they also have a process for catalytically upgrading that into jet fuel, and that's being deployed. But even within a single company, and I don't pretend to know their economics, like their techno-economic analysis and so forth, but if you switch to gasification of plant biomass, I would presume that the economics change quite a bit, as does the technology, because syngas from steel is probably of a different quality than steel mass from syngas from a plant, from like plant biomass. And so that's just one example, but that's one that's literally being deployed today. That's probably the best example on the biofuel side. Kind of thinking about the chemicals, there are certainly some chemicals that are already on the market today. Then the economics are going to be dictated by a lot of different things, including, is there a premium for renewable feedstocks instead of petroleum-based ones? For a long time, or for a little while, the answer was absolutely. People loved it. That was, I don't know, 15, 20 years ago. And then it became an emphatic no way. People are never going to pay for this. And then it's coming back around where a lot of people are willing to pay a premium for non-petroleum based products, especially in things like healthcare and makeup and things like that. So in the chemical space, yeah, there's just a lot of diversity, and there are going to be a lot of companies that crash and burn, and there'll be some that make it through and make a product. And yeah, I'm not sure I'm in a position to really say which are going to be which.
Another question here is, what will be necessary to make this practically and economically feasible to begin addressing the problem of highly mixed post-consumer plastic waste?
Yeah, so we have a paper that just came out in Science, so maybe a month or two ago now. So biology, I doubt that biology is going to be able to break down mixed plastics efficiently to make a bioprocess out of that. But there's a lot of synergy between chemical catalysis and biocatalysis. So chemical catalysis is good at a couple things. One thing it's really good at is breaking down things that are complex, but it makes a whole mixture of things. And chemistry is not good at dealing with that unless that mixture is inherently valuable, like maybe it's a great fuel or something. There's an old saying, what is it? It's something along the lines of, there's a world of difference between a valuable mixture of chemicals and a mixture of valuable chemicals. So like you can break a lot of these polymers and plant biomass into things where each individual component could be really valuable, but there's no way to separate them out in an economical way. So I actually forgot where I was going with all of this, but oh yeah, but like, you know, synergy between chemical processes and biological. Biology is really good at taking a mixture and making one thing out of it. And so that's the value that biology adds. And so if you imagine a process where you start with mixed plastics, all the plastics, you know, maybe you use chemical catalysis to break it down into components that microbes can eat. The microbes take that mixture and they make one thing out of it. Maybe that thing by itself is all ready to be used industrially as a solvent or a polymer building block or a fuel or whatever else. Or maybe chemistry is ready to take that molecule and make it into something else. You know, maybe you hydrogenate it or polymerize it or whatever else to make that kind of final product that you're interested in. Because chemistry is also really good at taking one thing and making it into one other thing. So biology is good at linking those two things together.
I think we've covered most of the general questions that were on the chat. Do we have any?
Do you think, or is anybody yet looking at the possibility of using these kinds of systems for treating, let's say, municipal wastewater? You know, where you get these personal care products and pharmaceuticals that are going into our wastewater treatment systems where it's not effective, that there's no good way to treat them, or they're just going out into the environment completely untreated.
Yeah, so first off, yes, people are interested in that and researching it. It's actually really hard just because there's so many different molecules and they're at such low concentrations. You know, they may be in high concentrations, you know, for a hormone, like if it's, you know, interfering with, you know, whatever, like human systems or frogs or whatever. But you know, for an organism trying to eat it, it's really a tiny concentration. And so, you know, I think that most people working in that space are working with natural communities and trying to, you know, maybe not so much make products out of it, but you know, definitely removal of carbon. Sometimes the cells themselves can be harvested for fish food or other things like that. So just because cells make protein and stuff that can be used as animal feed. So there are potential applications, but I think using engineered organisms for a lot of those trace molecules is going to be a really big challenge that may be insurmountable. But that doesn't mean that you know, there isn't space for research and you know, maybe phosphorus recovery. You know, phosphorus is a critical element that, you know, we're having less and less of, and farmers really need it. So can we recover phosphorus from wastewater, for instance, or, you know, critical materials, like the rare earth elements that are used in electronics and stuff. So it's not exactly wastewater, it's getting away from your original question, but, you know, can we use microbes to capture, you know, rare earth elements? And there's a lot of work going on in that space. And so, you know, thinking about how do we engineer microbial communities, how can we use natural microbial communities? These are all things that people are very interested in. Yep.
Are a lot of these bioprocesses you're talking about endothermic, that is, that requires significant energy to be added from the outside?
Typically, they're going to be exothermic. So for small molecules that are made by fermentation, it actually releases quite a bit of heat. So a lot of people don't know this, but like bourbon distilleries shut down in the hottest part of the summer to clean and, like, you know, everything. But it's because technologically, it's because the fermentation process produces heat, and it's enough heat that in the, you know, fall, winter, and spring, it can naturally dissipate that heat. But in the hottest parts of the summer, it just kills the yeast. And and they can't afford to have coolers running chillers running to cool their reactors. And so that's anaerobic stuff, but also aerobic stuff. Oxygen is a really good electron acceptor, releases a lot of heat, a lot of energy when doing respiration. And so, yeah, cooling is a bigger challenge rather than heating in most biotechnological processes.
Adam, there is one more question here. First of all, many people have thanked you for a stimulating presentation. A question that came from David Fields is, you spoke mostly about monoculture development and some two-stage processing. Are you also considering symbiotic cultures, for example, as found in sourdough bread?
Yeah. So we're doing a little bit of work in that space. The big challenge is it's hard to engineer organisms to play well together. So in almost all of these scenarios, we're hoping to make, 100 grams per liter of our product, for instance. So a lot of it. And typically it takes a fair bit of engineering to get a microbe to be able to tolerate that much of whatever the product is. And so if you have two members, now you have to engineer both of them to make it, or at least both of them to tolerate it. And maybe they have their own roles within the community, but it's challenging to engineer multiple organisms to play well together and to do well in a single bioprocess. There are people who are true believers in that approach, thinking that, well, natural communities do it that way, we should engineer microbes to do it that way as well. Their research is valid, but my perspective is that in most cases, if you're trying to make a single thing, it's typically going to be easier to engineer a single organism to do all of it rather than to engineer multiple organisms. If instead you're thinking about, well, wastewater treatment or anaerobic digestion, things like that, those are cases where, you're almost certainly going to need communities, and maybe you can kind of engineer it to do some of the things you want, or maybe you just kind of let it go to its natural endpoint and just I guess get as much out of the process as you can.
Well, it's now 1:30, and I think it's time that we all thank Adam for a wonderful presentation and a very good question and answer period.
Well, thanks.
Shut off the recording.